Well, good afternoon, everybody, and welcome to this Swift Analytical and CellDynamics webinar on what you'll see is some unique technology for the rapid and simultaneous determination of weight, size, and mass density of spheroids and organoids. And to give you an idea of how popular this is, it seems we've got over 300 delegates registered for this, which is brilliant. So people are just joining us quite rapidly at the moment, but we will continue because we appreciate time pressures. First of all, my name is Malcolm. I'm one of the Swift analytical team, and I'm going to be chairing the session this afternoon. There will be the usual opportunity for questions, which you can forward via the chat facility, and we'll try to answer as many as we can within the time available. Or of course, if you think of anything after the meeting, then please just contact Swift directly. Our main speaker today is Francesco Bacchi from CellDynamics. And Francesco's got an MSc in Industrial Biotechnology from the University of Turin in Italy. Then after 3 years of working in the lab, researching on isolation and the analysis of circulating tumor cells for cancer liquid biopsy, he joined CellDynamics as the application specialist. And his main field of expertise is in the implementation of standardized methodologies for the generation and analysis of 3D cell culture models, such as cancer spheroids or stem cell organoids. However, first I'd like to introduce Dr. Mebs Survey, who's the Managing Director of Swift Analytical. Mebs has got a PhD in analytical chemistry from the University of York, and he spent most of his career in establishing new technologies for blue chip and SME companies. He set up Swift in 2011, initially as a business development consultancy, but now it's a fully fledged UK distributor. So I'll first of all hand over to Mebs.
Thank you very much, Malcolm, for that introduction, and hope everybody's keeping safe and well. Just wanted to take this opportunity to thank everyone for giving up the time to join us today. We're living in very difficult and unpredictable times, but hopefully things are now coming to an end with the release of the vaccination programs. I just wanted to take this opportunity to those of you who don't know Swift Analytical to give you a very brief introduction to the company. As Malcolm mentioned, we started in February 2011. We celebrated our 10-year anniversary last month. Remotely with the team, but it was still very nice and very emotional. We're based in the beautiful historic city of York. And what we are aiming to do now that we are a fully fledged distributor is to bring technologies to scientists and researchers in the areas of single cell omics, high resolution printing, 3D cell culture, and high performance reagents to enable assay developments for drug discovery. So the first product I just wanted to introduce to you is the fluid cell biopen. Unfortunately, the animation isn't working on here today, but effectively what it is, it's a liquid handling system which allows you to use this biopen tip that you see here on your right-hand side. It comes as a complete system, which is a control box, a fluidic system, and a dispensing module which fits to a micromanipulator and to your conventional microscope. And what this allows us to do is to probe with great precision and accuracy single cells or groups of cells for applications such as assay development or single cell omics. The next device is high resolution single cell precision bioprinting, and this is possible with the BIOPHYSICS system. What we are able to do is to place individual cells into flat sheets. So for example, here, if we had the video running, we are able to demonstrate how liver cells can be surrounded by fibroblasts. And the biggest shift here is that we don't require any biogels. And because of the very low shear forces that are generated from the system, we are able to print very delicate systems such as neurons, stem cells, hepatocytes, and so on. what we're really keen on is to try to do as much as we can to reduce plastic waste. It's a problem which we all face within the lab, and we've helped to co-develop this product by Imrali Inventions. And effectively what it is, it's a cleaning system for plastic cell counter slides. So on the top of the image, you'll see the typical usage per lab in an average week. But what we can do is we can reduce this usage down by at least 50 times. And what we can do is we can wash, clean, dry, and recycle plastic cell counter slides in less than 20 seconds without affecting the workflow, but having a positive impact on the environment and also affording you great cost savings. Another application area that we are focusing on is in high-speed, high-viability dispensing of spheroids, cells, and organoids using our liquid handling system from BIOPHYSICS. We're able to dispense with high precision and high viability cells and organoids with multiple channels. So if we use a single type of cell or single type of organoid and we want to dispense it into a 1536 well plate, this can be done in less than 2 minutes. If you have the same cells in all eight dispensers, that cycle time is reduced significantly. We are also offering, and we're also aware that there's a big shortage of slides that are available, plates that are available for generating spheroids and organoids. And here we're proud to partner with Facilitate and Kugelmeyers to enable you to get an immediate supply of any plates that you need for cell culture. So looking at the if you are looking for single cell high throughput applications, then the Facilitate plates are perfect for your needs, and they are performing better than the Corning and the Thermo Fisher plates that are currently available on the market. If you're looking for higher throughput, so if you wanted to generate clusters of organoids or spheroids, then we recommend the Kugelmeyers plates. Our next slide is basically just a reminder that we are also able to generate COVID reagents and kits and various proteins for your assays. Again, these are in short supply generally, but we do have a ready stock. They're high performance. We've got some really fantastic endorsements, and they're ready available at a fraction of the cost. So I don't want to take any more of Francesco's time. Just wanted to say how excited we are to be partnering with CellDynamics. It's been a pleasure to work with these guys for the past few months, and especially in light of COVID and some of the challenges that they've faced. So I'd just like to congratulate them for bringing this groundbreaking product to market, and take this opportunity to hand over the floor to Francesco. So thank you for your attention.
Yeah, many thanks, Maps, for your introduction. Good afternoon to everyone. I'm really happy because there are tens of people attending this meeting today. I'm just trying to share my screen. Okay, so again, thank you for today's opportunity to disclose the features of our weight physical cytometer. And let's start with a brief introduction about CellDynamics. CellDynamics is a biotech company born in 2013 in Bologna. Most of us come from academia, and our main expertise regards the development of microfluidic devices for the analysis of 3D cell culture, especially 3D spheroids and organoids. And our technologies are hybrid because they integrate microfluidics with electronics and informatics. And today we will cut into details the features of the weight system that is, let me say, a lab weighing scale specifically designed not for liquids and powders as we normally do, but for 3D biological samples. We have already established a nice network of exclusive distributors to cover a large part of the world. So Swift Analytical is covering UK and Ireland. So feel free to reach out to us directly or to our local distributor for any more inquiries or requests. And now let's just move on to the reasons that pushed our R&D to develop this device. When we work with spheroids, but also with clusters, organoids, we can actually measure lots of parameters. For example, the dimension and the morphology with imaging techniques, but also the viability with biochemical assays. We can also quantify the expression profile with different methods. We can extract the DNA and run NGS analysis. But so far, there isn't a rapid and easy way to measure the physical parameters out of your 3D cell culture. And our system is going to answer to this need. And in addition, the spheroid formation is a multi-step process. It means that after the initial cell seeding, cells begin to aggregate each other, and after a delay period for the synthesis of the protein involved in the cell-cell junction, they form a compact 3D spheroid with tighter connection. So I know that most of the audience is really familiar with this kind of mechanism, but I'd like to put the focus on how these objects are evolving over time, also from the physical point of view. If we look at the picture on the left that is taken from a paper, we can see the images of spheroids cultured in U-bottom ultra-low attachment well plate at different time points. So basically, we have two parameters that are currently in use to classify these objects: the cell seeding number and the culture dates. Samples undergo a compaction phase from day 1 to day 2, and later on, they start increasing in size day by day, but this sample very fast reaches a plateau phase. So over 250 microns of diameter, the spheroid exposed 3 main layers: the necrotic core, the quiescent intermediate zone, and the outer proliferative ring. And only the cells at the periphery are doubling and viable. So these samples are not increasing so much in size if we pass from day 5 to day 13. And if we only look at the diameter, perhaps we are missing a part of the story, okay? So these samples are not increased so much in size, but they are getting darker. So something is changing. Probably the density, the weight, the thickness of these samples, we are not able to quantify this variation. But biophysics also compresses a biological relevance, because the size and the compactness of these spheroids influence the formation of gradients of oxygen and nutrients, and may also alter the drug penetration as well. So for all these reasons, measuring the physical parameters is worth the time and the effort. And as far as it concerns 3D bioprinting, one of the recent trends is to use stem cell derived spheroids as building blocks instead of cells embedded within nitrogen, as we do normally. And tissue spheroid can be assembled into macro tissue through this tissue fusion process. And new fabrication tools are under development to control this fusion of the spheroid through the direct placement of these spheroids in close proximity. But moreover, also new technology to characterize and standardize this spheroids production are needed. So basically, we know that 3D spheroids kiss as well, but under particular conditions. We need them viable, we need them compact, round, and possibly also with a diameter below 250 microns to prevent the formation of a necrotic core, and anything else. So if you have also any other suggestion, but these are more or less the basic condition. And in order to bioprint human organs, is necessary to develop technology for the production, but also for the standardization of spheroids. So this tissue spheroid must have standard size, standard shape, and also standard biophysical properties. For example, if we assume that the approximate volume of an average adult human kidney is 250 centimeter cubes, and we assume that the diameter of a tissue spheroid is 200 microns, we must fabricate 6.25 millions of tissue spheroids. And we need them to make fuse together. So far, we are just observing these processes. We are trying to bioprint larger tissue, but we cannot quantify the biophysics of these objects. We cannot discriminate the properties of these individual spheroids from the properties, biophysical properties of these bigger spheroids under fusion. And this is the solution that we are proposing. It's called weight. It's basically a physical cytometer. So it's the only fully automated instrument for the physical characterization of 3D sphere-like biological samples in terms of weight, size, and mass density. That is the ratio between the mass and the volume of the sample, and it is an index of the spheroid compactness. This technology is flow-based. It relies on a coupled gravimetric and dimensional analysis. The device is really small, so it can also be placed under the biological safety cabinet. Behind the safety door, we have the front side, and we have the weight chip compartment in the upper side that is designed to host our proprietary weight chip for the analysis of the samples. And below, we have the sample and liquid holder area that is designed to host 4 centrifuge tubes. Starting from the left, we have the vessel A, the first one, and the vessel B tube that are designed for collecting and sorting the target population of spheroids in the vessel A and the contaminants in the vessel B. And then we have the sample holder tube, that is the second tube from the left, that collects your suspension of spheroids and organoids under testing. The fourth tube is a 50 milliliter tube containing the weight analysis solution, that is our calibrated buffer for the analysis. And then we have the waste bottle and the 0.45 microns air filter for the air flushing in the instrument. So the device acts likewise a common flow cytometer. So it means that the suspension of spheroids is withdrawn from the sample holder tube by activating a peristaltic pump, and the spheroids are driven individually, so one by one, inside the specifically conceived weight chip. They are analyzed one by one with our bright field imaging setup in combination with a tracking algorithm. It means that our samples are carried just above the field of view of the camera, and in this particular moment, we stop the peristaltic pump and we let the spheroid fall down only by gravity force. So as such, the instrument tracks the spheroids during their free fall motion throughout the analysis channel with the buffer at rest. The camera takes images every 100 milliseconds to the single spheroid that is falling down, and the software converts the free fall speed of the spheroids in a mass density value based on a physical law that is Stokes' law. So it's basically sedimentation theory. And in addition, the weight software calculates the radius of the sample and approximate its shape to predict the sample volume. So we have the mass density, we measure the sample volume, and in the end, we can extract the weight of the sample in nanograms. This is the paper we have recently released in Micromachine Journal. It covers all the details of our theoretical approach and the basic principle of the device, so you can get the free full test access anytime you want for more information about the basic principle of the device. In brief, the weight system collects three outputs: the size, the weight, and the mass density. The device is calibrated with polystyrene beads, as we normally do in flow cytometry. So we have a sharp precision and accuracy that are higher than 99%. For what regards the working region, the system works with all sphere-like samples, can be spheroids, organoids, embryoid bodies, from 50 to 500 microns of diameter. Every sample is measured up to 10 times. It means that we can repeat 10 times the measurement on every single spheroid to get mean values and standard deviation. So this is done to increase the robustness of your data and, of course, to make more reliable the statistical analysis. One important function of the weight system is the sorting. So the system is able to sort and recover target 3D spheroids on the basis of custom ranges of sample diameter or sample mass density. And the user can also refine the data population from the outliers. So, for example, from poorly circular spheroids or loosely aggregate of cells. We can do that based on the standard deviation we have around our mean values. We set a cutoff, so the user can also refine the data afterwards. Our device represents a breakthrough, so it promotes this paradigm shift in in vitro research. So we are really familiar with this kind of workflow. We grow 3D cell culture, and then we expose the sample to drug solution, and typically we measure the in vitro efficacy with the variation of the size, so size shrinkage or in vitro viability assay. From now on, we can add also additional information in this context. For example, we can use the mass density as a marker of the spheroid compaction and compactness that can also be linked to the maturation process of the spheroid. And as regards the drug testing, these parameters can also be used as physical outputs, label-free outputs of in vitro drug efficacy. So, for example, we have already demonstrated that we can correlate the weight loss to the cell death of the cell within the spheroid, and also the diameter shrinkage and the mass density variation can be properly matched to the existing readouts coming from in vitro viability assay. So we are adding more information to what we already know, and we are doing that on a physical basis. Let's now take a look at the experimental workflow. In first instance, of course, we have the 3D cell culture. There are several methods to grow spheroids and organoids. The ultra-low attachment multi-well plate is the easiest way to do that. Today, we ask customers to introduce as a sample input from 20 to 100 spheroids for each test condition. And that's why micro-pattern plate or flat bottom plate are more compatible compared to U-bottom plate, because these two products create spheroids at a higher yields. The main drawback of flat bottom plates is that they create spheroids with heterogeneous size and shape. On the contrary, micro-pattern plate represent our most recommended solution because they can create homogeneously sized and shaped samples. For what regards the U-bottom plate, in that case, we have only one spheroid in every well. So the major pain point is that this kind of reagent asks a higher effort in pipetting to manually transfer and group spheroids from the well inside the single sample holder tube. We have already tested all these products, so our most recommended solution are these kind of plates, but you can, of course, still continue working with your suspension method for 3D cell culture generation. And after that, there is the sample loading. We group and transfer from 20 to 100 spheroids, depending on the sample, on the experiment need, for each test condition in the sample holder tube, and we will suspend the sample in 7 ml of weight analysis solution, our calibrated buffer in terms of viscosity and mass density. And now we are ready to set up the instrument. We can load the chip. We can, of course, refill the 50 ml tube with the weight analysis solution, and we are basically start to do the analysis and the sorting of our sample. Let's now take a little look more in depth around how our system works. The weight physical cytometer uses a proprietary software that is called Libra to operate. It is intended for research use only, so not for use in diagnostic procedure. The first screen is dedicated to the experiment settings. So at this stage, the operator can set the experiment name, the number of sample you want to analyze for each test condition, the robustness, so the number of test repetition for each sample, and the size range. This is a rough pre-selection of approximately the size of the spheroids we want to analyze. And after that, it is possible to proceed with the sorting options. So the operator can define custom, here we are. Yeah, here we are. So the operator can define custom ranges of diameter or mass density in order to recover and sort targeted subpopulation of spheroids in the vessel A. And now we are ready to start the experiment. The first procedure is the filling procedure. So we fill the entire network with weight analysis solution. We can also remove any bubble that may be present in the weight chip analysis channel. They can be removed at any time by clicking on pause button and then selecting the remove bubble function. That way we flushes air inside the weight chip, and then we fill again the chip with weight analysis solution. We can remove any bubble if present. And after the filling procedure is completed, we click on measure and we are ready to start the analysis. So the weight physical cytometer withdraws the sample from the sample holder tube and drives the spheroids inside the weight chip channel. If more than one spheroids are introduced inside the weight chip area, the system will automatically separate them. So only one sample will be analyzed, the first one in the row, and the other one are transported back to the sample holder tube. The selected spheroid is colored in red, and it is carried at the top of the weight chip channel that is the high quality camera. In the very first phase, we are estimating the weight of our sample in order to calculate the proper force of the peristaltic pump to push it upwards without any shear stress. You see that it's bouncing a little bit up and down. And then we have the centering phase. The spheroids is getting colored in yellow, and we are using the microcurrents created by the geometry of the channel to center the sample in the middle of the channel. And now we are ready to measure, okay? The diameter measured for a couple of times. We have selected this repetition inside this particular region of analysis. And after that, we can measure with the low quality camera the fall speed of the single spheroid in this other analysis region. Also in that case, we selected 2 repetitions. So we measure 2 times the samples that is falling down across the field of view, and we are able to measure the mass density. If any debris is present in the analysis chamber, we can of course remove impurities by clicking on pause and selecting the remove impurities function. And basically the device works automatically. The operator is just asked to control the quality of the sample he is analyzing, but everything is basically automated. We can have a quick overview of the results of our spheroids. So these graphs, diameter and density, are automatically updated throughout the experiment. And on the right side of the Libra software, we have the overview. So we have the timer of the experiment, we have information on the protocol that is running, and we have the information of all the samples that are collected in the vessel A or that are discarded in the vessel B. I would just like to put the focus on the sorting procedure. So after the measurement is completed, we can use this additional channel to sort and recover target population of spheroid. So after repeating the second measure, we can use this additional channel to recover the spheroid. This mechanism is flow-based, so we are only generating microcurrents to sort and recover in this additional channel these target spheroids that are connected to the vessel A. Once the experiments are completed, we can elaborate the data. So data are real-time exhibited in the software, but they can be easily exported in an Excel spreadsheet file. And we suggest the customer to show them as box and whiskers plot. This is the list of compatible samples that we have already measured in our lab. There are more or less 4 compatibility criteria for the analysis with our sample. Samples need to have a sample size included between 50 and 500 microns of diameter. We have to have sphere-like morphology. So the more circular the spheroid are, the better our software works. We also need to have a stable suspension of samples without significant aggregation in cluster. And of course, it would be better to have size uniformity related to that particular test condition of spheroids, okay? Just not to cover any possible statistically relevant differences between positive control and treated samples. And we have already tested a good number of tumor spheroids, stem cell spheroids, and also we are trying to analyze, I think in the next future also, airway organoids and colorectal cancer organoids. And let's now move to the application list. We have identified 4 basic area of application of our device. The first one is basic research. So we can use our device to provide a label-free, non-invasive method for the standardization of 3D cell culture methods. So to standardize and optimize protocols. And here we have some example. These are results taken from a paper that has already been published in Frontiers in Immunology. Here we have grown spheroids, mainly composed of 4 different colon carcinoma cell lines, HT29, SW620, DLD1, and HCT15. It seems that spheroids of HT29 from a preliminary bright field investigation have irregularly and rough shape, while the other 3 cell types create more round and compact spheroid. So this is really nice because we have created spheroids in flat bottom ultra low attachment plate, and we started from the same cell seeding number. But despite starting from the same cell seeding number, this cell line creates spheroids with different physical properties. These are the results taken with our device. We can see that SW620 and DLD1 generate spheroids with a higher weight, a bigger diameter, and a higher compactness, a higher mass density compared to HT29. So that way, our system is really consistent with the preliminary investigation and thoughts we have with the bright field analysis. HT29 are not so promising for the creation of this spheroid. They have heterogeneous size, they create smaller spheroid. And if we look at the trend of the mass density, we can see that these trends are preserved when we measure the number of cells composing the spheroids with confocal microscopy, or when we quantify the ATP content with cell titer glow kit from Promega. Also in that case, we have that SW620 and the DLD1 spheroids contain a higher number of cells and are more viable. So they have a higher quantity of ATP compared to HT29. So our data really match very well with the results coming also from our other kind of tests. And another point that can be interesting is for cell factories. So gathering precise information on the spheroid size, weight, and mass density value over time is also important when we want to scale up the process in cell factories. This is just a suggestion for everyone in the audience that is working with stem cell spheroids. Here we have cultured spheroids composed of keratinocytes for 6 days in agri-well plates from Stem Cell Technologies. And we have analyzed our spheroids at different time points, at different culture days, to see how they evolve over time. Starting from the second and third graph, we have observed similar trends in size and in weight. Passing from day 1 to day 2, we see a reduction in the size because spheroids are getting more compact. They are creating tighter connection, so they are aggregating. They are still aggregating. And from day 2 to day 6, they start increasing in size because cells are proliferating, are doubling, and indeed, sampling is increasing in weight as well. For what regards the mass density, the situation is really intriguing because we see the initial increase in the mass density that is consistent with the aggregation phase that we have seen before, but after, in a second phase, we have the decrease in the mass density. This is mainly due to the secretion of soluble factors by keratinocyte, for example, collagen, laminin, fibronectin, that can promote this kind of, let me say, social distancing. This is very famous nowadays among the cells. They are creating this physical distancing between the cells, and so we have the decrease in the mass density in the second phase after the first preliminary compaction phase. So all this data could be really relevant also for monitoring the growth kinetics of 3D cell culture. And along with the protocol standardization, these physical parameters coming out from the weight cytometer can also be used as quantitative outputs for the quantification of in vitro drug efficacy on a physical basis. Also, these results have been collected in a scientific manuscript that is under review, and the sample consists of colorectal cancer lovo cells that are grown in Euplesia cells, in Euplesia plates, sorry. This is a micropattern plate from Corning Life Science. Spheroids were cultured for 7 days, and then they were exposed for 24 hours to an anti-cancer compound that is an inhibitor of glucocorticoid receptor. And we tested live spheroid and then also fixed spheroid with our procedure of spheroid fixation with PFA 4%. With the preliminary investigation with bright field and confocal microscopy, we can see that spheroids look like more compact after drug exposure. It seems like if we look at the borders of the spheroid, they are more regular in treated sample compared to the positive control. Probably this is the effect of the drug exposure, since we have a collapse in our sample. And these qualitative results coming from the imaging are confirmed with the data coming from our weight cytometer. If we start from live spheroid, but we have the same trend also in fixed spheroid, we see that we have a reduction in the size, in the diameter, and a reduction in the weight passing from the positive control to the treated sample. And we also observe a good match with the reduction in the protein content. It means that our drug is promoting the cell death. So we have diameter shrinkage of the spheroid, and in the same time, we have the reduction in the weight due to cell apoptosis. What is really interesting regards the mass density. Treated samples show a statistically significant increase in the mass density compared to the positive control. And this is mainly due to this sample collapse. Since the weight and the size are simultaneously decreasing, this increase in mass density is mainly due to the fact that the volume is decreasing proportionally much more than the decreasing of the size, of the mass of the spheroid. So volume is decreasing much more than the mass, and in the end, the ratios of the mass density is significantly increasing. Okay, so this is really nice because basically we think about the spheroid disaggregation after drug exposure, but in this case, with this particular experiment, we have observed the spheroid collapse, and we can quantify this collapse as a significant increase in the mass density, in the compactness of the spheroid. We have repeated the same experiment with fixed spheroid, and we have preserved, we have kept all the statistically relevant differences. So this was an additional validation of our internal protocol for the spheroid fixation with PFA 4%. It means that if differences are present, we are sure that we can preserve this variation also after fixation. And this is very important for us in order to receive samples from worldwide and to offer remote demo in these COVID-19 times. The last application for today regards cell therapy. So beside the common drug testing with chemotherapeutic, the weight system can also be applied for evaluating the in vitro efficacy of cell-based therapies, as when, for example, we use immune cell killing activity to treat and kill tumors. These results are included in a paper that was released in Frontiers in Immunology last December in collaboration with a couple of immunologists here in Italy. And we first grow colorectal cancer spheroids for 6 days in flat bottom ultra low attachment well plate. And after that, we added NK cells in ratio 1:1 compared to the number of tumor cells composing the spheroid. And so we tested the in vitro killing efficacy of NK cells, and we tried to analyze this effect on a physical basis. We tested 2 exposure time, 6 hours and 24 hours, and we characterized the physical properties of our samples with the weight device. We tested 2 different cell lines, SW620 and DLD1. For both cell lines, we have observed a statistically significant decrease in the weight and in the size, both at 6 and 24 hours. This means that NK cells are promoting the cell death, and these results with the weight is also consistent with the decrease in the fraction of living cells that we have measured with commonly used in vitro viability assay. So we are able to match these results with what we normally do. Differences arises when we look at the mass density plots, also in that case. If we focus on SW620 spheroids, we see a slight increase in the mass density after 24 hours of exposure of NK cells compared to the untreated spheroids, compared to the positive control. But if we look at the graph of the mass density for DLD1 spheroids, we see that this increase is statistically relevant. Seems that after 24 hours of exposure to NK cells, we have a significant increase in the density of the spheroid after NK cell infiltration. So to investigate the reason behind this increase in the mass density, we perform confocal imaging on spheroids with the infiltrating NK cells that are labeled in green. And you can clearly see that we have a significantly higher number of NK cells that could infiltrate in the DLD1 spheroids throughout their surface. Instead, in SW620 spheroids, NK cells are only located and accumulated at the peripheral zone. So this different infiltration rate may explain the differences in mass density that we have detected with our flow-based device. And this can be useful because it can open to the study of a different mode of action of NK cells to attack and destroy the tumor, depending on the cell type composition of the spheroid, but also depending on the physical properties of the spheroid as well. So I would like to thank you very much for your time today. And many thanks for Matt and Malcolm for organizing this webinar. I saved the last 15 minutes for the question and answer session. So I'm really excited to hear your impression and your question. We would be happy also to offer free demo tests with our device. We can do that. We can work from remote due to this COVID-19 lockdown or restriction. So as I said before, we can offer that, or we can, of course, schedule an on-site visit when the restriction will be eased. Thank you very much.
Great, thank you, Francesco. That was brilliant. So thank you for that. And we do have some questions which have been coming in. The first one is from Bisan. I'm not sure whether you can see them, but the first question is: spheroids typically form uniform spheres. How does your system account for the heterogeneity of this shape of the organoids?
Yeah, so it depends on the cell culture methods you have. Generally, we have homogeneous spheroids, but in some cases we have also heterogeneous spheroids. Our system better predicts the shape and the volume of the spheroids where they are round. Okay, but we can of course also predict or estimate the volume of not perfectly circular spheroid, and the user can decide to refine the data under testing from, let me say, outlier, can be ellipsoid, can be poorly circular cell aggregate at any time during the experiment, based on the standard deviation around the mean values.
Mm-hmm. Great, thank you for that. The second question, which I think we know the answer because you have a button on the system for it, it says, does the system accurately work with spheroids extracted from hydrogels when the cells are grown or encapsulated in the hydrogels, then degraded for analysis because of the debris? And sometimes the debris is difficult to filter. Is the system affected by the debris?
Yeah, so the system works likewise a common flow cytometer, so it works on a suspension of spheroids. So the procedure is really streamlined when you grow spheroids in suspension. We have entered into discussion with a company that has already released on the market a product, commercially available solution for the safe harvesting and recovery of the organoids from the matrigel, for example, from matrigel domes. We know that generally a research group has their own protocols for the safe harvesting of organoids from the gel, but in that case, our idea and our effort is to standardize this procedure. So this is something we can do, and we would also interested in testing. So it's something we can manage.
Next one is from Magdalena.
So just for what regards the debris, if you remember, in the first screen of our software, you have the size range slider. It means that you can select with our device to analyze only, let me say, spheroids that are bigger than 100 microns or bigger than 50 microns. It means that all the debris or all the single cells that are undesired for the analysis cannot be recognized and detected from our software. So the management of the debris is quite easy because we can also have the function remove impurities. So it's not a problem at the moment for us.
Great. Now that was my understanding from the video there. Magdalena asks and said, great presentation, which we agree. Do you actually image your spheroids in the micro wells? And do you experience challenging regarding the shape, round or flat, of the well? But again, I think you've been explaining that.
We generally suggest the customer, of course, to monitor the growth in the micro well plates over time before sample loading in our device. This is a good question because there are companies that are building their core business around the geometry of the well plates. Yes, the geometry of the well plate can have an effect, especially when you work on stem cell spheroids. They can, unfortunately, somehow influence the stem cell differentiation. We have a partnership or collaboration with a company that delivers a good end product for the better preservation, let me say, of the stem cell-like phenotype of stem cell spheroids. We can talk privately about that.
Okay, thank you for that. Julia asks, could you give us more information about the speeds of sedimentation of the spheroids and the throughput of your of the method?
Yeah, so the system is able to measure 10 spheroids for every hour. So just to give a brief recap, we ask to load the spheroid related to each test condition from 20 spheroids from 20 to 100. This is more or less the range. And we suggest the customer to analyze at least 10 spheroids for every test condition to add, of course, robustness to the final data. We are able to analyze one test condition, so 1 tube, 10 spheroids for 1 hour. But it means that in 1 hour, we withdraw the sample from the tube, we analyze them with our device, and we also sort the spheroid in the vessel A. So it's an all-inclusive procedure that we can perform in 1 hour.
How does running them through the weight machine impact the organoid viability, and is O2 and temperature constant in the instrument?
Good question. For what regards the viability of the organoids, we are running tests. So I can update you, can keep you posted with the outcome of the results. What we know is our device is designed from the physical point of view, so for the geometry of the channel, and it is specifically conceived not to create any shear stress of the samples. It means that in the first preliminary phase, we do an estimation of the weight of the sample, be spheroids, organoids, not to push it upwards with too much force that can create any stress. So we know that we are really gentle with our device. We know that 1 hour in our salt solution probably does not decrease so much the viability of our organoids, but we need to carry out our final test before expressing this theory in numbers, but I can keep you posted. The idea of the device is to put it under the biological safety cabinet, and so of course also to work on live spheroids, live organoids. It can be also, it can be a little bit challenging, of course, also to analyze viable organoids, especially if you work with 400 or 500 microns of diameter, due to the limited penetration depth when you work with imaging techniques. So it's something that needs to be precisely measured.
We've got about 3 questions from Will. His first one says, have you considered the use of any other detection methods, such as dynamic light scattering, to help characterize and compensate for a heterogeneity? That's the first one.
Oh, yes. This is part of our patent, but basically the system works well with spheroids, also with clarified spheroids, because the detection mode is made in dynamics. So we compare consecutive images in bright field, and we are able to detect the object based on the gray levels of our samples compared to the background. The detection is image-based mode. Probably I cannot disclose any other information now, but I'm happy to discuss about that privately.
Great. And are you considering any solution for pre-fractionation of the samples?
Sorry, I missed the audio. For that?
Are you considering any solution for pre-fractionation of the samples?
For pre-fractionation, I don't know, because our main goal is to preserve them viable and keep them keep their integrity. Okay, so our system works with spheroids, not probably with after the disaggregation of spheroids is not basically the common flow cytometer. We are not thinking about that so far.
Right. And the next one is, seems 100 spheroids is the limit of input per sample. Are we developing a more scalable throughput for screening applications?
Yeah, so
too much away.
yeah, so it's a good question. The maximum number of 100 spheroids was done, of course, to better approach also a kind of research group that works on organoids, on few number of spheroids, because generally also this is quite expensive dealing with the growth factors and anything else. But the idea, of course, is to scale up the speed of the system, the number of spheroids that can be analyzed per hour in order to create an high throughput version of our device. This is something that we are working on and probably will be the second version of our device. This limitation of 100 spheroids is mainly due not to have more than one spheroid contextually in the analysis chamber. It's just like that. So the researcher can easily estimate, can easily count the spheroid organoid concentration per milliliter. We have the procedure to do that very easily under the inverted microscope and can prepare the dilution. I think this is also, let me say, cost saving for the cost of the sample preparation and the experiment.
Mm-hmm. And I think we've got time for one more. Are the spheroids destroyed by the system, and can I retrieve them back to culture after taking the measurements?
Oh, yeah. When every spheroid that passes in front of the field of view is generally sorted and recovered. Okay, so the targets, we select a range of diameter and mass density. We recover the target spheroid in the vessel A, and all the contaminants will be sorted and collected in the vessel B. So potentially every kind of sample can be recovered. If we have destroyed spheroid, we generally see that just before the analysis, once they are inside the weight chip. It never happened before that we have the destruction of the spheroid during the analysis, because we are working with very low fall speed and with very low pumping speed. If we have a destroyed spheroid, it depends the shape of the final spheroid we have. The system works well when we have the approximation of the samples to a perfect sphere. So the more circular the sample it is, the better the system it works.
Mm-hmm. Excellent. Well, I think we've just about run to our time limit there. So I'd just like to say, well, a big thanks to Francesco for an excellent presentation and for the feedback from all the participants. That really is appreciated. This has been recorded, so we will be allowing access to it later because of one or two questions have been on that. So the recording will be available, so you can go back over it. If there are any more questions, I say, which do come up after this, then do not hesitate to contact Swift. We'd be happy to answer them for you. And also, if you like a copy of the paper that was mentioned, again, get in touch and we'll make sure that you get a copy of it. So a big thanks to everyone. Thank you for taking the time out. We appreciate it. It is tricky. Everybody's busy these days, but greatly appreciated. So on behalf of Swift and CellDynamics, thank you very much indeed, and have a great afternoon. Bye for now. Bye.